All right, let's dive right into this. Have you ever stopped to actually think about a fundamental, completely mind-bending biological contradiction? Every single somatic cell in your body possesses an identical carbon copy of your DNA, but somehow a neuron in your brain functions in total, complete isolation from the mechanics of a skin cell on your arm. I mean, think about it. If your genome is just a static, unchangeable instruction manual, a set of permanent blueprints, why do we not grow teeth in our lungs or eyes on our fingertips? When looking into the source material for this explainer, the answer to that mystery completely shattered how I view genetics, because we have to look beyond the DNA sequence itself into this masterful, invisible choreography that proves your genetic code is absolutely not your fixed destiny. Welcome to the software of your biology.
To truly wrap our heads around this, we're going to break this down into 4 perfect chapters. 1. The code. 2. The environment. 3. The glitch. And 4. The update.
Chapter 1.
The code. So to solve the mystery of why identical hardware creates completely different cells, we really have to rethink the entire dynamic. We need to stop thinking of DNA as the only boss in town. DNA is just the hardware. It's a static blueprint. Sitting right on top of that hardware is the epigenome, the administrative software. And the epigenome acts as this massive series of dynamic biological toggle switches. It dictates exactly which genes are read and turned on, and which genes remain completely silenced. It's the interface that defines the identity and the functionality of every single cell in your organism. Basically, if your genome is the alphabet, the epigenome is the punctuation, the capitalization, and the paragraph breaks that actually give the whole story meaning.
Now, it turns out that one of the primary mechanisms for how this software actually operates is something called DNA methylation. This is where a methyl group, which is literally just a carbon atom bonded to 3 hydrogen atoms, is added directly onto the cytosine bases of your DNA molecule. And this acts exactly like a microscopic do not disturb sign. Imagine walking into a vast library. The exact information you want is inside a specific filing cabinet, but someone has placed a heavy iron padlock right on the drawer. The genetic data inside is perfectly intact, but the cellular machinery simply cannot open the cabinet to initiate protein synthesis. That gene is locked away.
Then we have the second major mechanism, which introduces an entirely new level of physical complexity: histone modification. See, your DNA is unbelievably long and super fragile, so it stays organized by wrapping itself tightly around proteins called histones. Think of histones as the physical spools for the thread of your genetic code. When these histones are chemically modified, they can either tighten their grip on the DNA or they can loosen it. Imagine a really long roll of garden hose. If you coil that hose incredibly tight and bind it up with thick rubber bands, you just cannot run water through it, right? The DNA is buried and inactive. But if you unspool it and you remove those bindings, the water flows freely. The gene is suddenly exposed, wide open, and totally ready to be transcribed.
And all of these mechanisms build into a beautifully continuous cycle. This regulatory infrastructure isn't just random. It is governed by highly efficient cellular enzymes known as writers, readers, and erasers. Writers apply the marks, readers interpret them to coordinate the activity, and erasers remove them to restore the gene. Imagine this invisible maintenance crew working around the clock, 24/7, making absolutely sure the right tools are out on the workbench while all the unnecessary equipment is safely put away in storage. Because without them, your cells would suffer from transcriptional noise. Every single gene would fire at once, causing immediate cellular collapse. Just as rests in a musical score create a beautiful melody instead of a chaotic wall of sound, your epigenome uses silence to create biological harmony.
Chapter 2. The environment.
Now this is where the science takes a truly staggering turn. We used to think this software was completely sealed off, but then we discovered transgenerational epigenetic inheritance. Back in 1944, during the Dutch hunger winter, mothers experienced severe caloric restriction. And decades later, researchers discovered that the children born to these mothers had significantly altered methylation patterns. Their bodies had literally been chemically primed in the womb for a world of severe scarcity. So when they were born into a post-war world of relative caloric abundance, their preset biological software clashed with reality. And this led to a much higher prevalence of obesity, cardiovascular disease, and diabetes. It is exactly like a software program written for a server with tiny memory limits. When you transfer it to a modern, high-powered computer, it still behaves as if resources are scarce, and it just throws constant error messages.
To really ground this, think of it like inheriting a car. The classic view of evolution said that only permanent genetic mutations drive adaptation. That's like being handed a brand new, structurally different car. But epigenetic inheritance is completely different. When you inherit epigenetic markers, you don't get a mutated car. You simply inherit the exact specific settings chosen by the previous driver. The rearview mirror is angled precisely to where they needed it, and the seat is reclined deeply to their preference.
Your ancestors' traumas and environments set the dials, and you inherited the configuration.
But the crazy thing is, it's not just catastrophic historical famines that alter these settings. Look at the subtle, low-level signals that constantly bombard our epigenetic landscape today. Chronic stress, sleep deprivation, the presence of endocrine-disrupting chemicals. When your body experiences chronic stress, it produces a relentless stream of cortisol. And this cortisol actually physically interferes with the enzymes responsible for histone modification. It subtly but permanently shifts your inflammatory genes from the off position to the on position.
So, why does this matter? Because over time, the long-term effect of those genes being stuck in the on position is profound. It leads to systemic, low-grade inflammation, which is the root of almost every single age-related disease we know of. It's vividly like leaving the central heater turned to absolute maximum in your house right in the middle of July. Sure, the house is still perfectly functional, and nothing is technically structurally broken, but the internal climate is heavily strained. That constant, unnatural heat is eventually going to cause the paint to peel and the wood floors to warp.
Chapter 3. The Glitch. So what happens when the software actually starts to break down? Well, this introduces the information theory of aging. As we age, researchers found that our cells literally begin to lose their ability to read their original epigenetic instructions correctly. This phenomenon is called stochastic drift. It's the random, unpredictable accumulation of epigenetic noise. Remember those tightly regulated toggle switches we talked about earlier? Well, over time, they start misfiring. Genes begin expressing themselves at the wrong time, in the wrong place, or with the wrong intensity. The horrifying reality here is that over time, due to this drift, a skin cell slowly loses the specific markers that tell it to be a skin cell. It literally forgets its own identity, drifting into a state of semi-functionality. Why does this happen? Well, life requires constant DNA repair. But to fix a piece of damaged DNA, the cell has to temporarily remove those epigenetic markers to actually reach the damage. Once the repair is done, it has to reinstall those markers. But every single time it does this, there's a tiny, microscopic risk of a reinstallation error. Think of it like making a photocopy, and then making a photocopy of that photocopy, and then another. With every single cellular repair, the image gets slightly blurrier. The crisp edges get fuzzier. The instructions become harder and harder to discern until eventually, what was once a clear set of biological instructions becomes a page of illegible grain noise. This epigenetic decay is actually so predictable that scientists can now measure it with staggering precision using what's called the Horvath clock. By looking at the methylation patterns at specific sites on your DNA, they can calculate your biological age, which might be vastly different from the chronological age on your driver's license. If your biological age is higher, it means your epigenetic architecture is rapidly deteriorating. Think of an old, heavily used map. The actual territory the map represents, which is your underlying DNA, hasn't changed at all. But the map itself has been folded and unfolded thousands of times. The creases are so deep and the ink is so incredibly smudged that you simply can't read the landmarks anymore.
But here is where this source material absolutely blows your mind. This process might not be a one-way street. Researchers have discovered a set of proteins called the Yamanaka factors, and when partially activated in adult cells, these factors act as a literal factory reset button. In experiments with mice, scientists have actually used these factors to clear out that accumulated noise, successfully restoring the function of damaged tissues and even reversing age-related vision loss. The genetic information wasn't permanently destroyed, it was just hidden behind corrupted caches and temporary files, and science is learning how to reboot the system.
Chapter 4. The Update. We are rapidly moving from just understanding these glitches to actively hacking the code. Now, you've likely heard of CRISPR, the technology used to cut and edit DNA. But cutting DNA comes with a massive risk of permanent off-target mutations. Epigenetic editing is entirely different. Instead of changing the hard-coded sequence, we are just delivering regulatory proteins to specific locations to flip the switches. It is the profound difference between permanently deleting a web page from the internet and simply hovering over the tab to mute the volume. One is a destructive, permanent deletion of data. The other is a safe, temporary, and entirely reversible adjustment of the signal.
The therapeutic implications here, especially for something like cancer, are incredibly hopeful. Cancer cells are highly deceptive. They often survive by placing heavy methyl padlocks on your body's natural tumor suppressor genes. Traditional chemotherapy fights this with a scorched earth strategy, indiscriminately bombing rapidly dividing cells and causing massive collateral damage. Epigenetic editing, however, targets those specific methyl-adding enzymes to precisely pick the locks. It hacks the rogue security system, removes the padlocks, and restores the cell's original administrative commands so it can seamlessly self-regulate. We just don't have to burn the building down anymore to stop the bad actors inside.
But to fully grasp this topic, we really have to look at the data regarding the broader societal impact of this science. Because if our environment physically writes itself into our genetic expression, then socioeconomic conditions literally rewrite our children's biological software. The research highlights a crystal-clear feedback loop. Poverty, systemic stress, and toxic environments induce epigenetic changes that physically diminish a person's cognitive and physical resilience. And as we learn from the transgenerational data, those exact vulnerabilities can then propagate directly to the next generation.
As the source text powerfully concludes, this data forces a rewrite of how we view human development. Public health policy is not just about avoiding immediate danger. It is about protecting the biological integrity of future generations. A child's health is the direct product of an unbroken chain of environmental signals stretching back through their ancestors. We are realizing that to optimize the human system, we have to optimize the societal inputs that program it.
Which leaves us with this final profound realization. Your biology is not a destiny written in stone. It is a live performance constantly being directed by your environment and your choices. You have the ultimate administrative access. As we master this regulatory layer, maximizing the signal-to-noise ratio of our genetic expression, we have to ask ourselves, knowing what we know now about the software of life, are you simply the victim of your hereditary baggage, or are you the active author of your biological potential? Thanks for joining me on this explainer. Keep learning, keep questioning, and I'll see you in the next one.